up we have, I believe we have Kaylee Byers presenting on the Vancouver Rat Project.
Yeah, I'm going to take it out.
Oh, do you want this one? Here, use this one.
Yeah? Amazing. And I can move? I can move around. I had a little too much coffee to stand in one place. Thanks so much for having me. Any opportunity to get up and talk about rats is really a great day for me. So today I'm going to talk to you a little bit about a recent project we've been working on with the Vancouver Rat Project. I'll give you a little bit of background information on the project and some of the more recent work we've been doing, which was in large part part of Michael Lee's master's project. So why rats? Why do we care about rats? Rats transmit a number of zoonotic pathogens, from viruses to bacteria to parasites, so lots of different things, and we'll talk about some of those things. They're also ubiquitous urban pests. They're on every continent except Antarctica, and they seem to be doing just fine. So here's a video that I quite like from some of our research in the Downtown Eastside. I think you might have to go up a little bit for the bar. There we go. Yeah, yes please. So there's nothing special about this day. It's just your average day and where the areas we were trapping. Keep your eye on the garbage can. Whoa. Yeah, they're doing great. There's some healthy rats. So we have lots of rats here in Vancouver, and they form close association with people. So it's not that they're around but you never see them. They're in our backyards. Some of them are in our houses. They do well because we do well. And Vancouver's Downtown Eastside is particularly concerning for the risk of disease spread from rats to people. And the reason for that is that Vancouver's Downtown Eastside, you may have individuals who are more likely to come into contact with rats because they're on the street. And then another aspect of that is that if that individual comes into contact with a diseased rat, they may also be more likely to become ill through, say, being immunocompromised from either poor nutrition or intravenous drug use. So we focus our research, to date, we've focused our research in the Downtown Eastside. And so prior to 2010, there was no research done on urban rats in Canada at all. It's still the only project in Canada on urban rats and one of the only ones worldwide. There's a few projects in the States and there's also one in Brazil. And recognizing that, the founder of the Vancouver Rat Project, Dr. Chelsea Himsworth, started the project to essentially determine the prevalence of and risk factors associated with rat-associated pathogens in Vancouver. And so as part of phase 1, they trapped rats in the Downtown Eastside. So this map is showing you city blocks that were trapped. Blocks with the hashes in it mean that there was trapping done there, but that no rats were actually caught. And the red circles are representing rats, and the size of those circles represent number of rats. So you can see some of our blocks, we have many caught rats, others not as many. If you're familiar with Vancouver, this is Maine and this is Hastings here, and Insight, like the safe injection site, is in this area. So some blocks, many rats, others not as many. Over the course of 1 year, they caught 725 rats from those 43 blocks. And the Vancouver Rat Project looked for a number of targeted pathogens, so those things that we know that rats can transmit to people. Yersinia pestis, that's the etiologic agent of the plague, the big bad that most people will think of when they think of rats. Bartonella, a bacterial pathogen that can be transmitted to people through rat fleas. So can the plague. So can Rickettsia typhi, which is the causative agent of murine typhus. All bacteria, Sulhantavirus, and Leptospira interrogans. Leptospira is one that we'll be talking about a bit more, but it's also a bacterial pathogen transmitted to people through rat urine. Delightful. And then in addition to these targeted pathogens, something that I think the Vancouver Rat Project did that was really quite amazing was they decided to also look at pathogens that rats could carry just by nature of their ecology. So not pathogens known to be carried by rats or pathogens that rats are the primary reservoir for, but other pathogens that they could come into contact with in their environment and potentially transmit or be a carrier for in the environment. And those included methicillin-resistant Staphylococcus aureus, so the superbug, Salmonella, E. coli, and Clostridium difficile. And what did they find? Rats carried many things. No plague, so woo. No murine typhus, no sole hantavirus, but Bartonellas. We had positive rats for Bartonella, Leptospira, and all of these potential associates, which is interesting because it demonstrates that rats can carry a number of different pathogens, and that could be concerning because when you have pathogens that can come into contact with each other within an organism, perhaps you could have or encourage the evolution of more virulent pathogens or antibiotic-resistant pathogens. So they can carry lots of different things. We like to refer to them as a mixing bowl. But in particular, Leptospira interrogans is the one I'll be focusing on for this talk. So globally, there are over 1 million cases of Leptospira annually. And for the most part, individuals may present with a fever-like illness. In severe cases, leptospirosis can progress to kidney failure or pulmonary hemorrhage. Right now, there are cases of leptospirosis in Puerto Rico after the flooding event from the hurricanes. So Leptospira bacteria shed in rat urine, and if you're wandering around and you come into contact with rat urine and you have cuts and abrasions, you can potentially become infected that way. Most mammals can carry Leptospira, but rats are the most common reservoirs in urban areas, and they can also shed the bacteria throughout their lives and show no signs of illness themselves. So one of the really interesting things about Leptospira carriage in the first phase of the rat project is that they found that not all rats, or the distribution of infection among rats, was clustered. So you can see here, so we've got positive rats in red, and then we've got negative rats in green. And so some blocks, this troublemaker block here, has many infected rats, whereas surrounding blocks may have no infected rats that were caught, which suggests that there is some clustering of pathogens. Another aspect of this is that leptocarriage was associated with dominance characteristics. So when we say dominance characteristics, we mean things like bite wounds, weight. So bigger rats with more bite wounds were more likely to have Leptospira than smaller rats without bite wounds. So some suggestion that Leptospira is transmitted among rats through, say, social interactions, and that there's this clustering. Clustering is interesting. There's not a lot of research done on rats, rat ecology in general, as I was saying, but they're not thought to move very much. They're thought to stay within about the space of a city block. So the general idea is if they're staying close to home with their relatives and their colonies, then they're just transmitting the pathogens amongst themselves and they're not spreading them between blocks. So this led us to two questions. The first is whether or not clustering of Leptospira was actually a result of having very little rat movement among blocks. And the second was, well, if we have clustering and it's because rats don't move, what happens when we disrupt rat colonies? And what could be more disruptive than pest control practices? So if you're taking individuals and you're removing them and you're killing them through pest control, could you potentially change dynamics of social structures in rat populations and change disease spread? Okay. So here's phase 2 of the Vancouver Rat Project and 8 months of my life. All the red circles are traps. We trapped essentially in the same areas that we did in the first round. And over the course of 8 months, we caught 780 rats, primarily Norway rats. Our traps are on the ground, so we would be preferentially targeting Norway rats in that way from 36 blocks. This is our mobile rat van lab that we drove around every single day. VV, that's what we called it, VV. We did a short documentary with the CBC called Keeping Canada Safe, so if you want to see us driving around collecting rats, you can do that. This is an infographic they did of our rat van that makes it look much tidier than I think you can see that it was. But essentially, we would drive around, we would check our traps for rats. Those rats would come to the back of the rat van where they would be put to sleep, and we would take samples and we would re-release rats because we were looking for movement and change in disease over time. So it was a capture, mark, recapture study. So this is the basic process. We would capture our rats. You would reach in and grab the rat by their tail, put them into what is essentially a glorified Tupperware, administer isoflurane to put the rat to sleep. They would go on the back of, in the back of the van where we would take blood samples, urine samples, fecal samples. That rat would then recover, and we would re-release that rat to the exact place that we caught it the first time so that we weren't changing their movement or putting them somewhere where they hadn't been before. So question 1, rat movement. What did rat movement look like over time? So out of those 780 rats, we recaught 130, which is actually pretty good. But we only saw 9 instances of rats moving between blocks, which could suggest that there is some strong site fidelity, which means rats aren't moving much between blocks. Capture, mark, recapture is flawed in that you can only see movement of individuals when they enter a trap. All the other movement, I can't see. So it could be that rats move a lot more between blocks than what we're actually seeing by the method that we used. We thought of that. So we put some GPS tags on rats. Here are 2 little GPS tagged rats. Here's another GPS tagged rat up here in the corner. We used a protocol approved by the veterinarians at the CCM at UBC. We used a veterinary adhesive and suturing technique that's been used previously in bats. And we had 2 tag types. One tag type was on 8 rats, so here's 3 city blocks here, and I'm just essentially showing you the location of tagged, GPS tagged rats. 8 of those tags we would have to recapture in order to download data, and 6 of those tags had remote download capabilities. So go back, a year to me wandering the blocks at 6 a.m. with a little handheld remote trying to download data from GPS tagged rats. What did we find? They didn't work. Surprise. Why didn't they work? So out of the 14 rats we tagged, we recaught 3 of them, and the tags had been removed. There seemed to be no effect on health, so weight stayed pretty much the same. There didn't seem to be any infection or scabbing at tag attachment site. 3 out of 14 is very much on par with our overall recapture rate. It could be that tags were removed immediately after rats left, either by themselves or their colony mates, or that when GPS tags were trying to take reads, rats were underneath structures or dumpsters or overhangs, and so it could have been obstructed. If you are interested in reading this paper, it is coming out in the Journal of Urban Ecology, so you can check that out there. Okay, so rats, based off what we've seen with capture, mark, recapture, don't seem to move much. So perhaps that's why we have clustering of pathogens. Question 2. We wanted to know whether or not a pest control intervention could change rat movement. So the general idea was that we would look at prevalence of Leptospira in rats before the intervention, during the intervention, and then after the intervention. So in all those blocks, there's 36 blocks, they were essentially in groups of 3. They looked like this. So we would have control blocks where we had no intervention, and then we would have our intervention sites. So in those 3 blocks, in the center block, we would enact a kill trapping protocol where we would remove those rats and euthanize them. So 2 weeks, we did capture, mark, recapture in all the blocks. The next 2 weeks, only in this block, we trapped and removed rats. All other blocks, we did capture, mark, recapture. And then we came back 6 weeks later and we did capture, mark, recapture in all the blocks and continued to look for Leptospira. So essentially we were asking, what are the odds that a rat was positive for Leptospira before the intervention and after? We also looked at MRSA, and I forgot to take it out of this slide, but I'll tell you about it also as we go. So for Leptospira, we took urine samples. It took us a while to figure this out, to be honest, how to get it. A good urine sample. The answer is putting the rat over top of a Tupperware container and waiting for them to urinate and collecting the urine that way. We should publish that. That was a lot of work. Okay, so overall Leptospira, we had 15% prevalence. And we found that the odds of being positive for Leptospira increased by 9.5 times in blocks where we had an intervention in compared to control blocks or the blocks on either side of the intervention, suggesting there might have been some kind of change by removing those rats to rat social structure or transmission patterns. We didn't see a change with MRSA. MRSA we believe to be environmentally transmitted and not based off of social interactions. But Leptospira, we did see an increase. And our hypothesis is that by removing individuals, that created a new social dynamic or hierarchy where individuals were coming into contact more often, fighting, biting, potentially transmitting the pathogen more than they had been previously. So in conclusion, we believe that minimal rat movement between blocks could lead to a clustering of pathogens, that human interventions can potentially disrupt social structure and movement patterns of animals, and that there could be implications for disease risk to people if we see a result in increase in zoonotic disease in rats, then that could actually impact potential disease risk to people. Now, do we know what the detected disease is in people in the Downtown Eastside for Leptospira currently? We don't. There is an ongoing study now looking at whether or not we've seen Leptospira in people. I don't know the results of that yet. But what I would essentially argue is that by focusing on detected disease, we miss the bigger picture, and we aren't able to focus on the ways to mitigate disease transmission. So we miss out on understanding the ecology of the disease within its population. If there are vectors involved, we miss that as well. And all of that's happening within a local and global environment. So by understanding the basis of our pyramid, we can hopefully get at and understand risk to people. And with that, I will take any questions you happen to have.
Okay, questions for Kaylee? Over here.
So as you know, there are hundreds of species of Leptospira. I've heard you or this presentation before, but I can't remember. How do you actually detect it? And are you able to look for differences between whatever you're identifying, different strains or subtypes?
So so this time around, we actually didn't go as far to strain. We can only say Leptospira positive or negative. But in the previous phase of the Vancouver Rat Project, Dr. Chelsea Hemsworth actually looked at, I think, 22 of those samples and typed them to Leptospira interrogans, and all were interrogans for rats. Whether or not there are other circulating strains, we don't know. We guess that those strains that we have this time around are Lepto interrogans.
Question over here.
Hi, thank you. Nice presentation. I'm following up with the Chelsea's work since it started. It's a great project and had lots of information came out from this. And our lab, we do human diagnostic for the whole province for Leptospira and other zoonotic diseases. But the interesting thing that I mentioned that we don't see any human cases of Leptospira from Downtown Eastside so far. And 15%, 14% positive is quite a high number of positive. And also, quite a few years back, there is a study done by BCCDC along with some health authorities that they look at like all the downtown restaurant, the condition in the kitchen, and they found like mouse and rats and cockroaches, many restaurants. And so there are some interaction, but still no human cases. But I know because Leptospira, most of the cases self-limiting, but then it might cause higher problem. But my question is that, do you or your whole group are thinking of doing some human serosurveillance? That is the main way that you can see the magnitude of like transferring from animal to human. I think that if we look for the disease, we may not be able to see it, or maybe all the diseases are very mild and nobody's care or whatever. So that's why we're not seeing it.
Yeah, so there is, Chelsea and Dr. David McVeigh have undertaken a serosurvey of Downtown Eastside residents, and I'm not sure of where they're at with that project yet. So I can't comment on whether or not they found any cases. I think that they did. I say quietly because I know there's a webinar going. But I'm not sure what the numbers are. So one other aspect we're also looking at is just that. So we see cases in rats, but if we're not seeing cases in people, is there actually a health risk? And one other aspect that we've taken on in the last year is to look at potential impacts of rats on mental health. So the idea that even if there could, let's say we don't have cases of Leptospira, but downtown Eastside residents perceive a risk of rats to their health, what are the potential health impacts of rats beyond just zoonosis? So there is a component that's a serosurvey. I don't know the results, and we're also looking at broader impacts of health effects on downtown Eastside residents.
Thank you. So I think most people would agree that the only good rat is a dead rat. How would you, how do your results inform a pest control strategy if knowing that by having an intervention, the odds of lepto could increase up to 9.5 times?
Right, so at the Vancouver Rat Project, we're really promoting the idea of ecological and integrated pest management. So the idea of decreasing resources in an environment to minimize the size of rat population. So as you mentioned, definitely in interacting with individuals, many people say, well, I'll just put out poison and I'll just kill them. But poisons are largely ineffective. We see that when you poison an area for rats, essentially what you do is you remove that population and then the rats from the surrounding blocks come in, and so you're back to your previous population within as little as 4 weeks, plus you can have impacts on disease spread. So we are currently working with the City of Vancouver to, one, have a rat surveillance program to look at rat populations over time, and also to work with pest control professionals to look at how we can minimize resources for rats in the environment, to not necessarily get rid of all of them, because I think that would be a very lofty goal, but can we minimize rat populations to an agreeable level where they're having minimal potential health impacts, but also impacts on infrastructure and economic impacts?
Wonderful. Next up, we have Natalie Prisajecki.